In the study of human evolution, certain discoveries mark turning points, moments when new evidence forces us to reevaluate long-held assumptions about our origins. The field of paleoanthropology is built on fragmentary clues, isolated teeth, partial mandibles, scattered postcranial bones. Rarely do we find individuals preserved in articulation, and rarer do we still uncover fossils that challenge the very structure of a hominin family tree. In 2008, in the Malapa region of South Africa, such a discovery occurred. What began as a routine survey of cave deposits quickly escalated into one of the most significant paleoanthropological finds of the 21st century. The specimens identified there, Australopithecus sediba, would become the focal point of scientific debate, not because it fit neatly into our evolutionary framework, but because it refused to. This documentary examines Australopithecus sediba through a strictly scientific lens. We will analyze evidence, the fieldwork, the anatomical data, the geological context, and the phylogenetic implications with the same rigor applied in academic research. Each section will explore a different dimension of the species, drawing on peer-reviewed studies, comparative anatomy, and the broader fossil record. The significance of Australopithecus sediba lies not in sensationalism, but in the precision of its data. The fossils recovered in Malapa represent some of the most complete early hominin skeletons ever discovered. They provide an unprecedented opportunity to examine morphology across the cranium, dentition, upper limbs, pelvis, and lower limbs in a single species. These remains allow researchers to test hypotheses about locomotion, diet, brain organization, and evolutionary relationships with a level of detail rarely possible in paleoanthropology. But Australopithecus sediba also raises questions. Its mixture of primitive and derived traits, a phenomenon known as mosaic evolution, complicates attempts to place it within the linear evolution sequence. Does it represent a transitional form between Australopithecus africanus and early Homo? Is it a side branch that evolved parallel to the lineage leading to Homo erectus? Or does it represent an independent experiment in hominin adaptation, one of several contemporaneous species exploring different evolutionary pathways? Throughout this documentary, we'll examine these questions systematically. We'll begin with the discovery itself, the geological setting, the field methods, and the initial analysis that brought Australopithecus sediba to the world's attention. We will then move through the anatomy in detail, comparing each region of the skeleton to other hominins. We will explore the taphonomy and preservation of the Malapa site, the environmental context in which sediba lived, and the dietary evidence that reveals its ecological niche. Finally, we will address the phylogenetic debates, evaluating the arguments for and against Australopithecus sediba as a potential ancestor to the genus Homo. This is not a story of speculation, but of evidence. It is a case study in how paleoanthropology advances. Through careful excavation, rigorous analysis, and the willingness to revise our understanding when new data demands it. Australopithecus sediba is more than a fossil species. It is a reminder that human evolution is not a straight line, but a complex branching process shaped by environmental pressures, anatomical constraints, and evolutionary experimentation. As we examine the data in depth, we will see how the species contributes more to a nuanced and scientifically grounded understanding of our origins. Let's dig into it. The discovery of Australopithecus sediba at Malapa in 2008 represents one of the most consequential field moments in modern paleoanthropology. To understand the scientific significance, we must examine the discovery not as a moment of luck, but as a culmination of methodological innovation, geological insight, and systematic fieldwork. This section provides detailed evidence based on the account of the Malapa discovery, the geological processes that shaped its site, the excavation protocols used, in the immediate scientific impact that followed. The Malapa site lies within the Cradle of Humankind, a UNESCO World Heritage region encompassing more than 400 square kilometers of fossil-bearing dolomite. The region has produced some of the most important hominin fossils ever discovered, including Australopithecus africanus in Sterkfontein, Paranthropus robustus in Kromdorei, in Swartkrans, and early Homo remains. The geological foundation of the cradle is Malmani dolomite, a carbonate rock formed during Neo-Archean era. Over millions of years, acidic groundwater dissolved the dolomite, creating an extensive network of caves, sinkholes, and vertical shafts. These karstic systems acted as natural traps, preserving remains of animals and occasionally hominids that fell into them. Malapa is one such system. Its formation involved vertical shafts created by dolomite dissolution, collapsing cave roofs that opened access to the surface, debris flows that rapidly buried organisms, low oxygen chambers that slowed decomposition, calcite flowstone that sealed sediment and allowed precise dating. This geological context is essential for understanding why Malapa produced such exceptionally preserved hominin remains. By the early 2000s, paleoanthropologist Berger had become increasingly interested in the possibility that many fossil-bearing caves in the cradle remained undiscovered. Traditional survey methods relied heavily on local knowledge and visible cave openings. Berger hypothesized numerous collapsed or partially buried sites had been overlooked. To this test, Berger adopted a novel approach for the time: systematic satellite-assisted surveying. Using Google Earth and handheld GPS devices, Berger and his team mapped potential cave locations based on vegetation anomalies, surface depressions, dolomite exposure, historical mining disturbances, and topographic irregularities. This method allowed them to identify dozens of previously unrecorded cave systems. Malapa was one of them. On August 15, 2008, Berger, his graduate students, and his 9-year-old son Matthew were surveying the Malapa area. The site appeared unremarkable at first glance, a shallow depression filled with loose breccia and mining debris. But as his team walked the perimeter, Matthew noticed a small fossil protruding from the loose block of breccia. The fossil was a clavicle, and its morphology was unmistakably hominin. Subsequent inspection revealed additional fragments including mandibular pieces, cranial vault fragments, postcranial elements. The team realized they had found a major fossil deposit. Excavation began immediately, following strict stratigraphic and taphonomic protocols. The Malapa excavation was conducted using a combination of traditional and modern techniques. The site's breccia, a cemented mixture of sediment, bone, and rock, required careful removing using geological hammers, dental picks, micro-chisels, consolidants to stabilize bone fragment, 3D mapping tools to record spatial relationship. Each fossil was documented in situ with GPS coordinates, photogrammetry, orientation measurements, stratigraphic position, sediment composition. Excavation revealed that the site's fossils were embedded in a single, well-defined depositional unit, including a rapid burial event rather than a long-term accumulation. The Malapa site produced one of the most complete early hominid assemblages ever discovered. The two primary individuals are MH1, the juvenile male, Carabo, estimated age 12 to 13 years from dental development, nearly complete cranium, mandible, partial vertebral column, ribs, and upper and lower limb elements. MH2, the adult female, included partial cranium, mandible, nearly complete hand, pelvis, and lower limb bones. Additional remains represent at least 4 other individuals. though these are more fragmentary. The completeness of MH1 and MH2 is extraordinary. In paleoanthropology, articulated skeletons are rare. Articulated Australopith skeletons are rarer still. Malapa provided not just isolated bones, but integrated anatomical regions, allowing researchers to study functional relationships between the body parts. Determining the age of Malapa fossils required multiple independent dating methods. The primary techniques included uranium-lead dating, applied to the flowstones that bracketed the fossils bearing breccia. Results 1.97 plus or minus 0.002 million years. Paleomagnetic analysis identified magnetic reversal signatures consistent with the Olduvai subchron. Biostratigraphy comparisons with associated fauna including Dinofelis, Meganterian, antelope species typical of early Pleistocene environments. All methods converged on an age approximately 1.98 million years. This places Australopithecus sediba at a critical evolutionary juncture, shortly after the appearance of Homo habilis and near emergence of Homo erectus. Taphonomic analysis suggests consistent scenario. Individuals fell into a vertical shaft approximately 5 to 10 meters deep. The shaft acted as a natural trap, similar to death traps documented at the sites of Sterkfontein. Carnivore activity was minimal, as indicated by the absence of tooth marks. A debris flow, likely triggered by heavy rainfall, swept bodies into the deeper chambers. Rapid burial in sediment protected the remains from scavengers and weathering. Low oxygen levels slowed decomposition. Calcite precipitation cemented the breccia, preserving bones in exceptional detail. This sequence explains the articulation of many skeletal elements and the preservation of delicate structures such as the hand bones of MH2. The Malapa assemblage includes a diverse range of fauna: antelopes, carnivores, rodents, birds, and reptiles. These remains provide environmental context. The presence of woodland-adapted species suggests that Malapa was surrounded by mixed woodland and grassland, with access to water sources and rocky outcrops. Pollen and phytolith analysis support the interpretation, indicating a landscape dominated by C3 vegetation, riparian woodlands, and seasonal grasslands. This aligns with the dietary evidence later recovered from Australopithecus sediba. In April 2010, Berger and colleagues published 2 landmark papers in Science. The description of Australopithecus sediba and the geological and taphonomic context of the Malapa site. These papers generated immediate international attention for several reasons. The fossils were exceptionally complete. The anatomy showed unique combination of primitive and derived traits. The species was dated to pivotal evolutionary period. And the findings challenged existing models of early Homo evolution. The scientific community responded with both enthusiasm and skepticism, a hallmark of healthy academic disclosure. The controversy centered on the claim that Australopithecus sediba might be a direct ancestor of the genus Homo. Critics argued that Homo habilis predated sediba by several 100,000 years. The sample size was small. Mosaic traits do not necessarily indicate ancestry. Supporters countered that morphology, not chronology alone, determines ancestry. The pelvic, dental, and hand features of sediba align closely with early Homo. The completeness of the fossils provides a more reliable data than fragmentary remains of earlier species. This debate continues today and will be explored in depth a little bit later. Since 2008, the Malapa site has remained an active research area. Ongoing work includes micro-CT scanning of fossils, virtual reconstruction of crushed bones, sediment micromorphology, isotopic analysis, and paleoenvironmental modeling. The site continues to yield new data, reinforcing its status as one of the most important hominid localities in Africa. The discovery of Australopithecus sediba was not a matter of chance, but the result of innovative survey methods, geological insight, rigorous excavation, and exceptional preservation conditions. The fossils recovered in Malapa were a rare window into early Pleistocene hominin anatomy, behavior, and environment. Their scientific impact has been profound, reshaping debates about the origins of the genus Homo and the complexity of early hominid evolution. Thanks for watching the first part of Australopithecus sediba. When all parts are recorded, I will combine them into one full-length documentary. The posting schedule may be changing slightly due to me finishing school and my last 2 classes being rather rigorous. I will still post daily shorts, but long-form content may be posted every couple weeks. If you have any questions about what we've covered today, feel free to drop a comment below and I'll get back to you with any information. As per usual, all links to research, sites, discoveries, and museums can be found in the information below. If you like what you've seen here, don't forget to like, comment, and subscribe for more deep dives here on Fossil Matrix.